Have you ever wondered how it's possible that our eyes don't produce hydrogen chloride the way our stomach does, even though every cell in the human body contains the exact same set of genes? It's due to a process called gene regulation that can turn genes on or off like a switch. An important epigenetic mechanism that plays a key role in gene regulation are histone post-translational modifications or histone PDMs, and that's what my research is about. The homeostasis of these histone PDMs is crucial and can make a difference between healthy and disease. As in my research and focusing on the disease part, we end up with the concept of toxico-epigenetics, which refers to any form of toxicity that is due to alterations in the epigenome, which in turn mediates protein expression and cellular phenotype. For example, under the influence of a component, and this can be anything. Just think of something you eat or drink, or a substance from the environment, pollution, and so on. Pre-existing histone PDMs may change, which may possibly lead to major consequences, such as deformation, but even cancer, autoimmune disorders, or neurological disorders. Additionally, we live in a time where we want to refine, reduce, and replace the use of laboratory animals as much as possible, also known as the 3R initiative. Especially reproductive toxicity assessment, because this is essentially the most animal-consuming area in drug development. So therefore, we had the idea to develop a human cell-based assay that is capable of tracking changes in multiple histone PDMs simultaneously in response to a treatment with a certain compound. As we wanted to do this untargeted, we relied on liquid chromatography coupled to mass spectrometry. In our pilot study, we treated human embryonic stem cells with 4 different concentrations of 10 different compounds, as shown on this slide. The results were captured in heat maps to visualize an increase, which is indicated by red, or a decrease, which is a blue color, in certain histone PDMs. By screening as much compounds as possible and storing all these results in a central database, it will finally be possible to cluster novel compounds with compounds with a known toxico-epigenetic effect based on the same trends in histone PDM changes. And in this way, it will finally be possible to classify compounds according to their potential toxicity in a given targeted cell type. In conclusion, toxico-epigenetic screening on histones is feasible, and integration of this epigenetic information in the field of toxicology is a promising addition that offers the opportunity to gain novel insights in toxicological phenomena. And in this way, our workflow can contribute to the safe development of drugs, but can also be important in the fields of environmental toxicity and food safety. And if you want to know more about our work, go and check out our latest preprint indicated with an arrow on this slide.